Testing. Okay, go ahead and jump in. All right, so today we're talking about muscle, largely skeletal muscle. And skeletal muscle is important for a couple of reasons. It allows us to perform body movements. We can move the bones by contracting muscle and applying force to the bones. We can use them to make facial expressions, speak, breathe, and swallow. We use muscles to maintain our body posture. So as we are sitting up in a chair, this requires, unless we're leaning back in a lazy boy, if we're sitting up, this requires some muscle to maintain our posture. They stabilize joints and maintain the body's position. Protection and support. So they package the internal organs and hold them in place, especially the abdominal muscles. The abdominal muscles serve as a wall, keeping the abdominal organs inside, whereas the thoracic cage keeps the thoracic organs in place. So also regulating the elimination of materials. So we have circular sphincters that control the passage of material through orifices. So yes, at both the anus and the inferior margin of the bladder, which we'll talk about in A and P2, both of these sphincters, we have round muscles, yes, called sphincters, and at both of those locations, there are actually two. There's an internal and an external sphincter. The internal sphincters are made of smooth muscle, but the external sphincters are made of skeletal muscle. And both of them, both the internal and the external, at both the bladder and the rectum, control the passage of urine and feces out of the body. All right. I got super in depth with that because when I see sphincter there, I think smooth muscle because the majority of sphincters in the digestive tract and in the body are going to be smooth muscle. But yes, there are two made of skeletal muscle. Uh, Good. So then heat production. Muscle contraction generates heat, and so that helps maintain our body temperature. All right, so there are a couple characteristics of skeletal muscle as a tissue. It is excitable, so it has the ability to respond to a stimulus by changing electrical membrane potential. We'll talk about how that happens today. Conductivity involves sending the electrical signal or the electrical change down the length of the muscle cell membrane. Contractility, of course, skeletal muscle can contract. So this happens when the filaments slide past each other. We'll talk about those filaments, the thick and thin, in just a little bit. Uh, and this is how muscles contract, sliding filament theory. Then we have extensibility, they can be stretched, and elasticity, which is the ability to return to the original length following a lengthening or shortening of muscle. So each skeletal muscle is an organ. Each anatomical muscle, we can call them also, is a muscle. So one example would be the pectoralis major muscle, what many people call the pecs, the latissimus dorsi muscle, um, which many people call the lats, or trapezius muscle, which many people call the traps. Each of those is an individual organ. So there are multiple types of tissues in a skeletal muscle, or rather in an anatomical muscle, working together. So we have skeletal muscle fibers. Those are skeletal muscle cells. We also have connective tissue surrounding the individual cells and then surrounding packages of cells, bundles of cells, holding it all together. We also have blood vessels and nerves. Blood vessels supply the muscle with oxygen and nutrients and then carry away waste. And the nerves, of course, control the contraction of skeletal muscle. Skeletal muscle never contracts unless it's stimulated to do so by a nerve. All right. Now you can contrast that with cardiac muscle, which is autorhythmic. In other words, cardiac muscle, the muscle of the heart wall, stimulates itself to contract. They have pacemaker cells inside the skeletal muscle, which controls the pace of the heart and signals the heart to contract all on its own. So muscle fibers, also known as muscle cells, well, the individual cells are also known as muscle fibers. They're bundled together, and that bundle is called a fascicle. The whole muscle, the whole anatomical muscle, contains many fascicles, and a fascicle is made up of many muscle fibers. All right, so the muscle fiber, as we said, is the muscle cell. These cells are very long, though. They may not be very wide, but they're very long. They run the entire length of the anatomical muscle. So if you look at your biceps brachii muscle, which is commonly known as the biceps muscle, that may run the length of, uh, depending on the length of your arm, it may be, I don't know, 8 to 12 inches long. And so the individual muscle cells in that muscle, in that anatomical muscle, are each going to be about 8 to 12 inches long. They're going to run the entire length of the muscle. All right, now there are some connective tissue layers surrounding the muscle. Surrounding the anatomical muscle, that is the large one that we would call by name, such as the biceps brachii, the trapezius, the latissimus dorsi. Surrounding that is the epimysium. So remember, epi means above, and myo refers to muscle. So the epimysium is above the muscle, or in other words, it's the outermost layer surrounding the muscle. All right, and it's a layer of dense, irregular connective tissue that wraps the entire muscle. The perimysium is also made of dense, irregular connective tissue, and it wraps around the fascicles. Remember, the fascicles are bundles of muscle fibers, and the perimysium will house many blood vessels and nerves. Then lastly, we have the endomysium, which is areolar connective tissue that surrounds each individual muscle fiber. So it's a delicate layer that electrically insulates the individual muscle fibers, but it also supports capillaries, that is blood supply, and the binding of neighboring cells. Remember, with loose areolar connective tissue, there's um, relatively little protein in the matrix, mostly ground substance, and this allows plenty of space for blood vessels, and so loose areolar connective tissue tends to have a rich blood supply, and it's good for supporting other tissues, nearby tissues, such as the muscle cells. Now we look at the picture here. This is looks like the biceps brachii muscle, and the anatomical muscle is the entire muscle. Okay, the one that has a fancy name, fancy Latin name, biceps brachii, and it is composed of multiple fascicles. So if you look at here, we have multiple bundles, okay, of muscle fibers. All right, there's one big one sticking out here in the cartoon drawing. Each of these bundles of muscle fibers is called a fascicle. And then within the fascicle, we see multiple individual muscle fibers. All right, now if you're following along in the cartoon, you look at this individual muscle fiber. You know this is an individual muscle cell. This is one cell. And we're looking at the cross section, and there are all these other little circles in it. What's that? Well, those are the myofibrils. Those are the proteins that are actually going to cause contraction in the muscle, and we'll talk about those very shortly. All right, so the connective tissue layers, remember, surrounding the entire anatomical muscle is the epimysium. Epi means above. All right, deep to that is the perimysium, which surrounds the fascicle and kind of bundles all those muscle fibers together. Peri means around. That can be a little confusing because epi's above, peri's around, but I mean they're both around the muscle, right? But epi is the outermost, epi means above, peri is around, peri bundles, surrounds the fascicle, and then surrounding the individual muscle fiber is the endomysium, which is composed of the loose areolar connective tissue, which has a pretty good uh, blood supply. All right, good. And then the anatomical muscle leads up here to we've got the tendon, and we find the origin of the biceps brachii muscle uh, up here superior to the humerus, the head of the humerus. And the tendon actually passes through the intertubercular sulcus between the greater and the lesser tubercle there. All right, now the insertion is actually going to be down on the radius, and so when this muscle contracts, it's going to lift the radius. All right, good, good, good. So more connective tissue components. These are attachments for the muscle to bone. Remember, attaching muscle to bone involves a tendon, which is a cord-like sheet of dense regular connective tissue, or an aponeurosis we didn't mention before. This is like a flat tendon. It's a sheet of dense irregular connective tissue. An aponeurosis is like a tendon that's in instead of a cord-like form, it's in a sheet. It's flat and broad and thin. Then we have the deep fascia, which is composed of dense irregular connective tissue, and it's superficial to the epimysium. It's uh, above or outside the epimysium, and it separates individual muscles and binds muscles that have similar functions together. Then we have the superficial fascia, which is superficial to the deep fascia. It's composed of areolar and adipose connective tissue, and it separates the muscles from the skin. When I used to teach just anatomy lab, like not anatomy and physiology, but just anatomy, we would dissect cats, and we spent about a month on that because you can go through all the organ systems, all everything in depth. We're learning the anatomy. It's an anatomy class, so we would do that. And when you would separate the skin from, say, the abdominal muscle, what you would get is this sort of cottony, spiderweb-like material that had a very distinct sound as you pulled it away, and that was the superficial fascia. The superficial fascia, I share this story because I'll never forget it. It's very, very vivid. But uh, the superficial fascia um, is there connecting the muscle to the skin and the hypodermis. All right, blood vessels and nerves. So the skeletal muscle is vascularized and it has an extensive uh, network of blood vessels. Uh, Obviously, blood vessels are for the delivery of oxygen and nutrients and removing of waste products such as CO2, right? And then skeletal muscle is innervated by somatic neurons. When we talk about the nervous system, we'll talk about the somatic and the autonomic nervous systems. The somatic nervous system, just in short, is the portion of the nervous system that we're consciously aware of, those sensations, and then the muscles that we can consciously control, which is skeletal muscle. That is, the nervous system, the somatic nervous system, consists of nervous fibers receiving inputs that we're consciously aware of, and those nerve fibers that control skeletal muscle consciously. The autonomic nervous system, on the other hand, receives information from the digestive organs and things like that, and then um, sends signals through other fibers that will control the functions of just for instance, abdominal organs. If that was confusing, we will talk about the nervous system in depth later. Don't necessarily need to know all that, but you'll see the word somatic come up again. Somatic neurons, somatic nervous system. It'll come up again in the nervous system. All right, so the axons of the neurons are they branch at the terminal end, and they terminate or end at the neuromuscular junctions. The neuromuscular junction is a junction between a neuron and the muscle fiber that it innervates. All right, and this can allow for voluntary control of contraction. Skeletal muscle actually, as we said, never contracts unless it's stimulated to do so by the nervous system. If you want to be specific, that would be efferent fibers of the somatic nervous system. That last part will make sense when we get to the nervous system. All right, so parts of a cell or muscle fiber, we have the sarcoplasm. That's the name for the cytoplasm of a muscle cell. We said myo means muscle. That's Latin. Sarco means flesh and also muscle. Sarco is Greek. Okay. The word sarcophagus is actually Greek and it means flesh box. It's like an ancient coffin. Sarcophagus. Anyway, sarcoplasm. Here we're talking about the cytoplasm of sarco muscle cells. And they have the typical organelles in addition to contractile proteins such as the myofibrils, well, actually, specifically the myofibrils. All right, and so it has a specialized name. That is, it is a name to differentiate it from cytoplasm because it's uh, functionally different from cytoplasm. Sarcoplasm is the cytoplasm of a muscle cell, and it's different because it is largely filled with myofibrils. All right. Now, skeletal muscle cells can be multinucleated. They can have multiple nuclei. And the reason for that is if you look at your biceps brachii, we said these cells can be 8 to 10 or 8 to 12 inches long. And so one nucleus is not going to generate enough. Well, so if you have a nucleus at one end of that, it would take forever, maybe days for mRNA or for new proteins to reach the other end of that cell. I mean, it'd take a long time to travel down there through the sarcoplasm. Um, so to remedy that, we have multiple nuclei distributed throughout the muscle cell, throughout its length, so that we can generate plenty of mRNA locally throughout the protein fiber, or rather, sorry, locally throughout the muscle fiber, and generate new proteins that way. All right, the sarcolemma. Sarcolemma is the plasma membrane of a muscle cell. Sarco again means flesh. Lemma is going to refer to the plasma membrane. So this is the plasma membrane of muscle cells. Now, they are also specialized. They have a unique structure, and so they get a special name, sarcolemma. They have T-tubules, known as transverse tubules, and these are deep invaginations of the membrane. In other words, the membrane will fold in on itself, and this invagination or this infolding will travel deep into the interior of the muscle cell. We'll see a picture of that in just a little bit. That's important because we need to excite muscle cells and we need to convey that electrical signal down the length of the membrane. But they're also somewhat thick as far as cells go, and so we need to get that electrical signal deep into the muscle cell. And so as the electrical signal travels down the length of the muscle fiber, it also needs to travel down these two T-tubules into the core, you could say, of the muscle fiber in order to initiate contraction. All right. So the T-tubules, which are a continuation of the sarcolemma, they have voltage-gated ion channels. Um, and so when they open up, they will depolarize the cell. They will flip the polarity of the membrane, and that opens up calcium channels that are going to initiate contraction of muscle cells. All right, so here we see an individual myofiber composed of many myofibrils. All right, so if you look here, this is a myofibril, and these are the rope-like or cord-like protein structures that are going to be responsible for contraction. We can see that there's a lot of them in an individual muscle cell. Okay, an individual myofiber is made up of multiple myofibrils. We can also see there are multiple nuclei in the cell, and this is just a short length of it throughout the entire length of it, you'll find, excuse me, many nuclei. All right. Now, the outer membrane here is known as the sarcolemma, again, and you see these pores here. Those would be connected to the green structures. Maybe a little hard to see. It's kind of small. In fact, if you look at this bottom image, it's just a little bit bigger. They would be connected to these green structures. Those are continuations of the sarcolemma, all right, known as T tubules or transverse tubules. And so basically, when the electrical signal travels down the length of the sarcolemma, it's going to travel also down the T tubules, which are part of the sarcolemma, deep into the muscle cell. All right, and that depolarization, that flipping of polarity, is going to open up some calcium channels. All right, and those calcium channels are going to activate calcium channels on the um, terminal cisternae, which are these structures here in blue. Okay, and that will travel uh, down the length here, and then that releases calcium into the muscle cell, and then calcium initiates contraction, and we'll talk about how that happens in just a little bit. Here we see depolarization, and so normally we have a situation where the inside of the cell is more negative than the outside of the cell. We have the sodium potassium ATPase, or the sodium potassium pump, which is constantly pumping out sodium, all right, and pumping in potassium. Now they both have positive charges. Okay, so you may be thinking, how are we going to create a difference in voltage across the membrane if I'm pumping out positive ions and I'm pumping in positive ions? Well, first, there are a lot of proteins in the cell, especially on the inner surface of the membrane, which gives it a slight negative charge, just to begin with. And then we're also going to be pumping out 3 sodiums for every 2 potassiums we bring in. And so in this exchange, we're basically pumping out 3 positive charges for every 2 we bring in. Right? It's like that phrase, 2 steps forward, 1 step back, but at least you're moving forward a little bit, right? And this sodium potassium pump, there are several of them in the membrane, and they are constantly functioning to pump sodium out and potassium in. And so we build up a high concentration of sodium outside and potassium inside. And over time, what will happen is, yes, we will develop a relatively positive charge on the extracellular side of the membrane and a relatively negative charge on the inside of the membrane, the intracellular side of the membrane. And depending on the cell type, this charge is going to be different. In muscle cells, the potential across the membrane, the difference in charge across the membrane is about negative 90 millivolts. In neurons, it's going to be about negative 70. All right, so that'll matter later, not so much now. What does matter now is basically we also have these voltage-gated sodium and potassium channels. And when we depolarize the membrane, we're going to open the voltage-gated sodium channels, and that's going to bring sodium into the cell. Well, if the cell is negative inside and we start bringing in positive charges, what's going to happen to the charge? Right, it's going to become more positive inside the cell. And this increase in positivity, or the cell becoming less negative, is another way to say that, causes the potassium channels to open, and the potassium channels are going to allow potassium to leave. And so if we now have positive charges leaving the cell, it will become more negative again. All right, so let's go through the steps again. When we open voltage-gated sodium channels, and they open because there's been an increase in the voltage across the membrane, right? That's why we call them voltage gated. When the voltage increases, that causes the channel to change shape and basically open up, and then let sodium ions, which are at high concentration outside the cell, diffuse in. All right. So just to take it back to the beginning, when this opens up, when the voltage change causes the channel to open up, sodium ions will diffuse along their concentration gradient or down their concentration gradient into the cell. That makes the inside of the cell more positive, and we call that process depolarization. We have essentially flipped the charge across the membrane where it's now more positive inside the cell. It's less negative inside the cell. That change in voltage causes the potassium channels to open up. And then potassium, you remember, we've been pumping it into the cell, so it's at high concentration in here, and it's going to diffuse down its concentration gradient out of the cell. And when that happens, when we start letting positive charges flow out of the cell, potassium is positively charged, so we let positive charges flow out of the cell, that's going to make the cell more negative again, and we say it is repolarizing when it becomes more negative inside the cell. All right, that all of that is basically step one. I know that was kind of complicated, but the thing is, when the cell becomes more positive in this region, all right, that is actually going to open up sodium channels down the line. All right, that's going to open up sodium channels over here, and that's going to cause sodium to flood into this region. And then potassium channels down here will open and potassium floods out. And that's going to cause, again, down the line, more sodium channels to open up. We're going to continue this wave of depolarization down the length of the membrane as long as there's sodium outside the cell and there are voltage gated potassium channels in the next area over. And this process is called an action potential, where we depolarize one region, and then that causes the next region to depolarize, and then the next region depolarizes. And so, one more way to think about this is we've flipped the polarity of the membrane, which causes the next region to flip its polarity or depolarize, which causes the next region to flip its polarity or depolarize. And so again, when an action potential propagates down the membrane, when it spreads down the membrane, when it's carried down the membrane, that's that electrical signal we've been talking about, where a cell's plasma membrane, or the sarcolemma specifically here, carries that electrical charge, that electrical signal down the membrane. That's the action potential, where we are depolarizing this region, which causes the next region to depolarize, which causes the next region to depolarize. That's an action potential. All right. Good. So, one important thing here about the image that we're seeing. So you see how the membrane kind of bends and moves down into the cell here? That's a T-tubule. Okay, and you can see on the other side here also the membrane is kind of bent down. All right, and this invagination, or this infolding of the membrane, is called a T-tubule. And by continuing a small section of the membrane down into the cell, we can propagate or continue that action potential, that electrical signal, down into the cell along the length of these blue structures called the terminal cisternae. Now, the terminal cisternae have calcium channels. Basically, both the T-tubules and the terminal cisternae have calcium channels that work together in tandem. And the actual process is somewhat complicated, so we won't get into it. But basically, when the action potential travels down the T-tubule, that causes the calcium channels to open, and calcium floods out of the terminal cisternae, out of the endoplasmic reticulum, into the sarcoplasm, and then calcium is going to be the release of calcium is going to be the first step in actual muscle contraction. And so what we're talking about so far is stimulation of the muscle cell and carrying that electrical signal down the membrane. At the end of the day, what happens is this electrical signal, the action potential, causes calcium to be released, and then calcium release is the first step of contraction. All right, so myofibrils we talked about. Those are the long bundles of myofilaments, the long rope-like proteins that are going to be contracting, doing the actual work of contraction. Sarcoplasmic reticulum we talked about. Oh, that's sarcoplasmic reticulum. Actually, we didn't talk about. So if we go back here, I didn't use the right term earlier necessarily. So next to the T-tubule is a large, it's pictured blue here. Of course, in real life it wouldn't be blue, it would be clear. But in blue here is the terminal cisternae, which is an enlargement of the sarcoplasmic reticulum. And the sarcoplasmic reticulum is a modified version of the endoplasmic reticulum. The endoplasmic reticulum normally houses calcium ions. This one has been specialized to hold lots of calcium ions, and so it releases calcium into the sarcoplasm to initiate contraction. All right, so the two cisternae, we saw there's one on either side of a T-tubule. Those three structures together, a cisternae, a T-tubule, and another cisternae, are known as the triad. In cardiac muscle, we see the dyad, where there's one T-tubule and one terminal cisternae. That's in cardiac muscle. The triad's in skeletal muscle. All right. All right. So the sarcoplasmic reticulum, we said it contains calcium pumps. Well, we said it contains calcium. It contains calcium pumps that import calcium, and that calcium binds to calmodulin and calsequestrin. Basically, when we pump calcium into the sarcoplasmic reticulum to store it, eventually the concentration is going to rise to a point where it becomes difficult to pump more in. So calcium then binds to proteins that are in the sarcoplasmic reticulum called calmodulin and calsequestrin. And because they bind to those proteins, they're kind of pulled out of the solution, which means now we can pump more calcium into that space. And then we have calcium release channels in the sarcoplasmic reticulum, which are of course going to be voltage gated calcium channels that release calcium into the sarcoplasm. All right, so you can see that here we've got the triad, which consists of a terminal cisterna, T-tubule, and another terminal cisterna. This is in skeletal muscle. In cardiac muscle, it's the T-tubule and a single terminal cisterna. These processes work the same in cardiac muscle, but because they have gap junctions, you see less sarcoplasmic reticulum. It's less developed because there's less need for it. Ions can travel through the gap junctions. It's also autorhythmic, which means it's not stimulated by the nervous system. It stimulates itself to contract. Cardiac muscle does. All right, inside the terminal cisternae, we find these proteins, calsequestrin and calmodulin, and again, they bind to calcium. You see these individual sort of pearl-looking things, these individual dots floating around. Those are supposed to be free calcium. And there's a limit to how much free calcium we can have in the sarcoplasmic reticulum or the terminal cisternae, which are part of the sarcoplasmic reticulum. There's a limit to how much free calcium we can have in here. And so these proteins, calmodulin and calsequestrin, they bind it And if calcium is bound to these proteins, then it's not free calcium, and so we can pump more calcium in. All right, so the myofilaments, these are contractile proteins in the myofibrils, and there are two types, the thick filaments and the thin filaments. The thick filaments have bundles of the protein called myosin, and myosin looks like two golf clubs that have been wrapped around each other. They got this golf club shaped head, and of course, there are going to be two heads at the end of the protein. Then we have the thin filaments, which consist of bundles of a couple of different proteins. They're twisted strands of F-actin, and then F-actin is made up of G-actin units. Basically, F-actin stands for filamentous actin, and G-actin stands for globular actin. And all this is telling you is that one F-actin strand, F-actin is a strand of G-actin units. You can see it in a picture in a second. It'll make more sense then. G-actin has myosin binding sites where the myosin heads attach, and then we also see tropomyosin and troponin there, and they are regulatory proteins. So if you look down here, just so we can clear up the F-actin thing, F-actin or filamentous actin would be this long chain. In fact, there are two here, two wrapped long chains of these red dots. Each individual red dot is G-actin or globular actin. F-actin is the filament version of actin, composed of many G-actins. Good. So up top we have a thick filament, and it is composed of many myosin molecules. An individual myosin molecule looks like this, where we have two tails wrapped around each other, and then two heads up here. And again, I think it looks like someone took two golf clubs and just wrapped them around each other, and then at the end you've got this head, this myosin head. And the myosin head has an ATP binding site. It also has an actin binding site because it's going to bind to G-actin. All right. Good. Thick filaments. Now the thin filaments down below are composed of actin or F-actin, and it's going to be surrounded by this green rope-like protein called tropomyosin, and then attached to that is troponin, this blue protein. All right. So basically, G-actin has these myosin binding sites, and the heads of the myosin molecules will bind to the myosin binding sites on the G-actin monomers, or G-actin little balls here. All right, and so the head, it reaches across, it binds, basically it grabs on, and then it's going to pull. All right, when it pulls, it's going to, it's like tug of war where you're pulling a rope toward you. All right, so the myofilaments are arranged in repeating units called a sarcomere. The sarcomere is the unit of contraction. Contraction occurs at the level of the sarcomere. And it's composed of overlapping thick and thin filaments. And a sarcomere runs from one Z disc to the next. We'll see that in a picture in a second. But it runs from one Z disc to the next. The Z disc is composed of specialized proteins that are perpendicular to the myofilaments, meaning they run in the opposite direction. So if the myofilaments run lengthwise, the Z discs could be said to run horizontally, right? And they're basically anchors for the thin filaments. Then we have, so the positions of the thin and thick filaments give rise to alternating I and A bands. The I bands will appear light and the A bands will appear dark. And the easy way to remember that is that there's an I in light and there's an A in dark. And where you see the A band, I'm sorry, where you see the I band, you're only going to see thin filaments, and that's why it's light, is because you're only seeing thin filaments there. But in the A band, that's where you're seeing thick and thin filaments overlap, and so it's going to appear darker. Because we have these alternating bands, the I bands and the A bands, the light bands and the dark bands, that gives us the appearance of striation, which is why they call skeletal muscle striated muscle. Also, cardiac muscle has I bands and A bands, and so it also is striated muscle. All right, so the H zone and the M line, which are at the center of the sarcomere, will be found in the A band. All right, so here we see a sarcomere. You've got a Z disc and a Z disc, and the sarcomere runs from one Z disc to the next. And you can see from the lines how these filaments run the length of the sarcomere, and then running perpendicular to that is the Z disc. The Z disc serves as an anchor for the thin filaments. All right, in the middle you've got the H zone, which is where you will find only thick filaments. Now it's part of the A band, but only thick filaments are found in the H zone. And the M line, this is the M is a German name. I can't remember. It's like Meischer or something like that. But I just remember it as M middle. The M line is in the middle. That's fair enough. All right, and that's the attachment site for the thick filaments. That's where they're anchored, is at the M line. Now we zoom in here. Okay, we're going to zoom in to one sarcomere, just from one Z disc to the next one. All right, so you got one Z disc, okay. At the M line in the center, you find that's where the thick filaments made up of myosin are anchored. At the Z disc, all right, you find that that's where the thin filaments are anchored. All right. And you also find this protein connectin, which is going to sort of stabilize the position of the thick filaments as well. All right, so they're trying to show you here the A band covers the region where you're going to find thick filaments or thick filaments and thin filaments. The H zone is the region where you only find thick filaments. And then the I band is this region where you only find basically the thin filaments by themselves. If we go back, you can see that alternating pattern of light and dark. Around the Z disc is the I band, where we only find the thin filaments. Then we find the A band, okay, through here, which is going to be pretty dark, and it's composed of the thick and thin filaments. The H zone's a little bit lighter in the middle because there's only thick filaments here, no thin filaments. And then we reach another I band at the other Z disc composed only of thin filaments. And this alternating pattern of I band, A band, I band, light, dark, light gives us the appearance of striation. All right, so now we're looking at some individual proteins here. We got myosin in the thick filament, and they have an actin binding site. They're going to reach across, bind to actin, then the head is going to pull on the actin, pull on the thin filament, like you might pull on a rope, right? Like in a game of tug of war, you pull on a rope. The myosin heads are going to be pulling the rope toward the M line. All right. Good. All right. All right, so we mentioned connectin, spring-like, and it helps connect the Z disc to the thick filaments and stabilize them. Dystrophin is another important protein. It anchors some of the myofibrils to the sarcolemma, to the plasma membrane. And the protein's named dystrophin because an abnormal version of this protein, a mutant version of this protein, is responsible for the disease muscular dystrophy. In the disease muscular dystrophy, specifically Duchenne's muscular dystrophy, the most common type, there's a non-functional version of dystrophin that is coded by the patient's genome, their DNA. Um, there's been a mutation that makes the protein non-functional, and because it's non-functional, the myofibrils will tear away from the sarcolemma, will separate from the sarcolemma, and that actually causes the muscle cell to break down over time, and so you see a wasting of muscle. Oh, here we go. Duchenne muscular dystrophy. We just described this. So problems begin in early childhood. I'm sorry, I'm not laughing at the disease. Obviously, I'm laughing at the that I forgot this slide was here and went ahead and talked about it, which happens somewhat frequently. All right, so the problems begin in early childhood, as we, well, have not yet covered. We covered the rest of this. You'll see problems with walking as well as muscle atrophy. The muscle will waste away, and you'll see postural issues. Unfortunately, it is uncurable, and patients rarely live beyond age 30. All right, so microscopic anatomy of the skeletal muscle. We also see a lot of mitochondria in the muscle. Mitochondria are important for producing energy, of course. You know, we've said it before. You can go through glycolysis in the cytosol of a cell, and you produce 2 ATP for every molecule of glucose you break down. That's not a lot, you know, but you can do it without oxygen, but it's not a lot. It'll keep you going for a couple minutes maybe, but just a couple. Um, so if we have mitochondria in a cell, and we do in all of our cells except for red blood cells, um, maybe a couple others like platelets, although platelets aren't cells. Anyway, almost all the cells have mitochondria, and what mitochondria are going to do is perform a process called oxidative phosphorylation, where we take the leftover byproducts of glycolysis and we're going to generate another 30, 32, 34 ATP from the waste products of glycolysis, and that requires oxygen, and it's going to generate CO2. So, you know, taking the waste products of glycolysis and making another 30, 32, 34 ATP is a big deal. Uh, Now, so textbooks disagree on exactly what number of ATP is made by the mitochondria through oxidative phosphorylation, and they disagree because it's not a perfect process, right? Like it's it involves the sequestration of hydrogen ions, and there's a little bit of leak, and so it's not totally efficient. It's not 100% efficient. And so ideally, we would get like 36, 38 ATP out of the whole thing. But the real number winds up being something like 32, 34 because of hydrogen ion leak. Without getting too into it, just textbooks don't totally agree on what number, but you know, it's in that area, 32, 34 ATP generated through oxidative phosphorylation. Um, all right, good. And so the reason we bring up mitochondria here, if almost every cell has mitochondria, why mention it? Muscle fibers are going to have a lot of mitochondria because muscle contraction, as you might guess, is going to require a lot of energy, a lot more energy than most other cells in the body will require for their function. And so we've got a lot of mitochondria in the muscle cells so that we can generate tons of ATP for aerobic exercise. Myoglobin is a protein that we find in muscle cells. You're familiar with hemoglobin, which is found in red blood cells. Hemoglobin binds oxygen and carries it throughout the circulatory system. And then whenever the red blood cell reaches a tissue that has low oxygen saturation, the oxygen will fall off of hemoglobin and then diffuse into that tissue. It's a pretty cool system. right, so why do we mention myoglobin? Myo again means muscle. This is a specialized uh, globin protein that is related to hemoglobin that is found in muscle cells. Basically, a long time ago, there was one globin gene, and then it got duplicated in our genome, and then over time, the two different versions have had time to mutate away from the original form. Now we got two variant forms, hemoglobin and myoglobin. Myoglobin's similar in that it also binds oxygen, but what it does is it binds oxygen in the muscle cell and it holds on to it until that oxygen is needed. And so if oxygen in the muscle cell is plentiful, myoglobin keeps holding on to oxygen. But if you suddenly start working out, you suddenly start running, and you use up the oxygen that's in the muscle cell and you can't replace it fast enough with the circulatory system, myoglobin will release its oxygen so that the muscle cell can do a little bit more work, a little bit more oxidative phosphorylation or ATP production. All right, you're also going to find glycogen. So there are only two organs or two tissue types which hold on to glycogen that store and use it, the liver and skeletal muscle. So skeletal muscle holds on to glycogen, which is the branched storage form of glucose. And whenever you begin to exercise for more than a couple minutes, you will start to break down glycogen in the muscle cells so that you can release glucose into the sarcoplasm, and then you can go through uh, glycolysis and oxidative phosphorylation and basically generate a whole bunch of ATP during exercise. It's an energy storage in form that we break down during exercise in skeletal muscle. As I said, the liver also produces glycogen when blood sugar is high, stores it away in glycogen. When our blood sugar is low, the liver will break down glycogen and share it. It will send it to the bloodstream, share it with all the tissues of the body. Skeletal muscle, on the other hand, is selfish, and when it breaks down glycogen, it's not sharing that glucose with anybody. Only that muscle gets to use the glucose that comes out of that glycogen. Liver's a little more giving. It shares that glucose with everybody. Then we have creatinine phosphate. Creatinine is a well, I think that might be a typo. Creatine's from the breakdown of creatine. Creatine phosphate holds on to a phosphate group, and basically we can quickly transfer that phosphate group onto a DP to recharge it and make it ATP again. When we break down ATP, it turns into ADP, adenosine diphosphate, because it's lost one of its phosphate groups. Creatine phosphate shares its phosphate group with ADP to make it ATP, adenosine triphosphate again. And so it's there to quickly recharge the ATP. All right. All right, so a motor unit is a motor neuron and all the muscle fibers that it controls. A motor neuron basically is a neuron that's going to control muscle, or doesn't have to be muscle necessarily, but in this unit it will be. And so the motor neuron innervates muscle, it controls muscle, tells it to contract. The motor unit consists of that neuron and all of the fibers that it stimulates. And so we see two motor neurons here, one in blue, one in red, and then we see a bundle or a number of muscle fibers. And you can see that the red neuron innervates this muscle fiber, this one, this one, this one. The blue one innervates these two muscle fibers. All right. And so when the blue neuron, motor neuron 1, is stimulated, it will stimulate only these two muscle fibers to contract, and that is a motor unit. All right. The neuron, motor neuron 1, and these blue myofibers. And then motor neuron 2, the red one, and all of the red myofibers, that's a separate motor unit. So the motor unit again is the motor neuron and the fibers that are innervated by that motor neuron. All right, so the axons of the motor neurons come from the spinal cord or brain, and they innervate numerous muscle fibers. The number of fibers a neuron can innervate will vary. Small motor units have less than 5 muscle fibers, which allows for precise control of force output. Large motor units have thousands of muscle fibers, and they allow for the production of large amounts of force, but not precise control. And so small motor units will be useful for things like fine motor control, delicate touch, things you might do with your fingertips. Whereas large motor units will be involved in muscles like the quadriceps femoris, which helps us stand up from a squatting or a seated position. We need to generate a lot of force to do that. And so large motor units will be involved in standing as compared to uh, writing your name in the fog on a window, right? That would be small motor units. Or touching a cell phone screen, playing Candy Crush. There we go. Small motor units. All right, so the fibers of a motor unit are dispersed throughout the muscle, and they're not just in one clustered compartment. Basically, they don't all have to be touching each other. The neuron has to touch the muscle fibers, but the muscle fibers can be spread out throughout the muscle. All right, so next up is whoa, did we skip one? I don't know. Anyway, we got the neuromuscular junction next. We're talking about the synaptic knob, and this structure actually has a whole bunch of names depending on your textbook. It could be synaptic knob, uh, terminal end knob, or bulb, or a lot of physiologists seem to like the word bouton, terminal bouton. Synaptic knob's fine. Most people will know what you mean when you say synaptic knob. If you want to get real fancy with people, though, you can say bouton. They love that. This is the expanded tip of the neuron's axon, the motor neuron's axon, and it houses synaptic vesicles. Remember, vesicles are membrane-bound sacs inside the cell, and synaptic vesicles are going to be found in the synaptic knob near the junction with the muscle fiber, and they're going to be filled with neurotransmitters. Now, that's true of almost the entire nervous system, but specifically when we are talking about muscle, the neurons that innervate muscle will always use a neurotransmitter called acetylcholine. Other uh, other other synaptic knobs in the nervous system may use other neurotransmitters, such as, like norepinephrine or noradrenaline or something like that. But here, when we're talking about skeletal muscle, it'll always be the neurotransmitter acetylcholine that gets released. And specifically, well, no, we won't get into that just yet. Sorry, there's a lot of information here, and I'm struggling to try and cover it in a way that makes sense, cover it in an order that makes sense without trying to talk about things that come later. All right, so so there's calcium pumps in the synaptic knob, and they're going to establish the calcium gradient. Basically, we're pumping calcium out of the neuron, but there are also voltage-gated calcium channels. And so when the neuron gets depolarized, that increase in charge inside the cell will open the voltage-gated channels, and that causes calcium to move in. It floods in along its concentration gradient, so that once calcium's in, it binds to the vesicles and encourages them to fuse with the membrane and dump their contents out. And of course, their contents are acetylcholine, at least at the neuromuscular junction. The motor end plate is a specialized region of the sarcolemma. It has numerous folds and many acetylcholine receptors. Specifically, there are two types. There are two types of acetylcholine receptors, and so specifically, you'll find the nicotinic acetylcholine receptors. Nicotine overdose stimulates the nicotinic acetylcholine receptors. They're named nicotinic receptors because nicotine also binds to them. And so that causes muscle cells to depolarize more often and basically get excited. And then there's later a depressor phase, which makes it harder to do things like breathe. Nicotine, in fact, plants, well, tobacco plants, evolved to produce nicotine as an insecticide. Bugs that try to eat tobacco plants asphyxiate and die. So anyway, the good. How much of this we've covered? All right. The synaptic cleft is the space between the motor end plate and the synaptic knob or terminal bulb or whatever you want to call it. All right. So it's a narrow fluid-filled space, and when the synaptic vesicles fuse to the membrane of the synaptic knob in the neuron, they then get dumped out into the synaptic cleft, which is a narrow fluid-filled space, and they have to diffuse across this space to reach acetylcholine receptors on the motor end plate. Okay? In the synaptic cleft, we're going to find a protein called acetylcholine esterase. And acetylcholine esterase breaks down acetylcholine molecules. Acetylcholine, often abbreviated ACH, by the way. All right, and so here we see three neuromuscular junctions where the synaptic knob, this broadened region at the end of the axon, is sort of in contact with the cell. There's actually a space between them, but where it's going to stimulate the cell. The neuromuscular junction is the junction between the synaptic knob and the muscle cell. All right, and you can see here that there are many vesicles in the synaptic knob, and these vesicles, these are membrane-bound sacs filled with the neurotransmitter acetylcholine. So when the action potential travels down the axon, all right, it's going to travel down these branched pathways as well. All right, this is the electrical signal, the action potential. It travels down the axon. And then we're going to look here at this synaptic knob. In this region, we find voltage-gated calcium channels, and the change in voltage that comes with this action potential is going to open up those voltage-gated calcium channels. Then calcium floods in. There's a whole bunch of it outside the cell, and that's going to flood in now that we've opened up those channels due to the voltage change. And that's going to bind to proteins on the outside of the vesicles, which encourages them to bind to and fuse with the membrane of the synaptic knob. When they do that, they're going to dump their contents out into this thin space, thin fluid-filled space, called the synaptic cleft. And again, that's just the space between the motor end plate and the synaptic knob. Synaptic cleft is just the space between them, and we dump our neurotransmitter out into this space. Now the motor end plate is this specialized region of the membrane which has a lot of folds on it, and the folds basically increase the surface area. All right, and we're going to have a whole bunch of acetylcholine receptors on this portion of the membrane. There are going to be a whole bunch on the motor end plate. And when the acetylcholine diffuses across this space and binds to acetylcholine receptors, if enough of it binds, that's going to start a whole new action potential along the sarcolemma. And that new action potential travels down the length of the sarcolemma, and it travels down those T tubules, and it travels next to the terminal cisternae, and it causes them to release their to open up their calcium channels and release calcium into the muscle cell. And then, as we said, the release of calcium is the first step in contraction. Everything else we've talked about so far is excitation, neurons exciting muscle. Contraction starts with the release of calcium. Here you can see that again, maybe a little closer. We have the calcium pumps that pump calcium out constantly, and by doing that, we generate a concentration gradient where there's a whole bunch of calcium outside of the synaptic knob. When the action potential comes, that depolarizes the membrane here and causes these voltage-gated calcium channels to open. They open, and then calcium floods in along its concentration gradient or down its concentration gradient, binds to proteins on the vesicles, causes them to fuse, dumps out their contents into the synaptic cleft, and then the acetylcholine diffuses over and binds to acetylcholine receptors. Now, I should actually say acetylcholine receptors, these nicotinic acetylcholine receptors, are also ion channels, right? And so when we bind acetylcholine to the acetylcholine receptor, we're going to open up this ion channel. Now, it actually allows both sodium and potassium through. Normally, channels are specific for sodium, potassium, or calcium, and this one is non-specific. It'll allow sodium in and potassium out, just not necessarily at the same time. All right, and then, the last thing, we have acetylcholine esterase in the synaptic cleft. And basically, after acetylcholine is bound to the receptors, it gets released. The acetylcholine esterase finds it and cuts it up, breaks it up, and then the remaining parts get reabsorbed by the synaptic knob, so they can get recycled to make new acetylcholine and new vesicles full of acetylcholine. All right. So as we mentioned, the resting membrane potential is around negative 90 millivolts for the muscle cell. And what that means is that the inside of the cell is about 90 millivolts more negative than the outside, right? The difference in charge between the two sides, inside and out, is about 90 millivolts. It's more negative inside. So we say the resting membrane potential, the membrane potential, or the difference in charge across the membrane, is about negative 90 millivolts at rest. Just when it's not being excited or stimulated, just sitting there, the charge is going to be about negative 90 millivolts. All right. And this is established by the sodium-potassium pumps, which are constantly pumping sodium out and potassium in. However, if we let those run forever on their own, eventually, we'd get away from negative 90 millivolts. We'd get more and more negative. And so we have leak channels, which are special channels in the membrane that slowly allow some of the ions through. We have sodium leak channels and we have potassium leak channels that'll allow some sodium just a little bit back in and some potassium just a little bit back out. All right, so the leak channels keep us from getting too negative. All right. So we talked about this. Again, when the acetylcholine binds the acetylcholine receptors, these are ion channels that will allow sodium in because sodium's at high concentration outside the cell and potassium out. Now, in order to start an action potential, we have to reach a threshold, and so we need to make the inside of the membrane slightly more positive. And if we don't reach the threshold, then we're not going to initiate an action potential. However, if we do make the membrane more positive enough to open sodium channels, voltage-gated sodium channels down the line, then we will initiate an action potential, and that electrical signal will travel down the length of the sarcolemma, down the T tubules, and then stimulate the release of calcium into the sarcoplasmic reticulum. Now, calcium, we said, is the first step in contraction. All right, and so calcium is going to bind to the regulator molecule, the regulator protein on the thin filaments known as troponin. All right. So, wow, we've talked about all this already. All right. Here's a good picture again. Steps in excitation at the neuromuscular junction. All right. Myasthenia gravis is an interesting autoimmune disease. It's primarily seen in women. It's Latin for severe muscle weakness. And here we see that antibodies are produced that actually bind to the acetylcholine receptors at the neuromuscular junction. And the receptors, they get removed from the muscle sarcolemma through endocytosis. And because we remove receptors from the motor end plates, right, there are going to be few receptors there. And fewer receptors actually means it's harder to get enough sodium ions into the cell to generate a positive enough membrane potential to start an action potential, right? It gets harder to reach threshold because there aren't enough receptors available to get stimulated. All right. And if there aren't enough muscle, if there aren't enough acetylcholine receptors to get stimulated, then it becomes hard to excite or, well, excite the membrane and then initiate contraction. All right. Now, this actually says rapid fatigue is a symptom, and that's not actually true. Fatigue is not a symptom of myasthenia gravis. These people are not tired necessarily. They do experience what seems like muscle weakness because they're not actually able to stimulate the muscle. It's not that they're tired necessarily, and it's not necessarily even that the muscles are weak, but it's an inability to stimulate the muscle in the first place. The first muscles involved are eye and facial muscles. In fact, the first symptom many times will be droopiness in the eyes, droopy eyelids. And it can be followed by swallowing problems and limb weakness, and then in extreme cases, it actually can affect respiration, and it can be fatal if you're not able to breathe. All right. There actually are treatments available for myasthenia gravis, and they actually involve disrupting acetylcholinesterase or inhibiting acetylcholinesterase. Now, if you do that, what's going to happen is if you don't break down the acetylcholine, it spends more time in the synaptic cleft, right? If you're not breaking it down, there's nothing to destroy it or remove it. It's going to spend more time here in the synaptic cleft, which means it gets more opportunities to stimulate the few receptors that are remaining. All right, good. So moving forward, stimulation of the fiber is coupled to the sliding filaments. The coupling includes the end plate potential, which is the change in voltage here at the motor end plate, as well as muscle action potential and the release of calcium from the sarcoplasmic reticulum. The idea of excitation contraction coupling is that basically the neuron excites the muscle fiber, all right, at the motor end plate, and then can generate an action potential which excites the muscle fiber and carries its electrical signal down the way. And then we release calcium, and then calcium initiates contraction, and so contraction does not occur without the calcium release, which does not occur without the excitation. All right. All right, so an end plate potential. So the acetylcholine receptors are chemically gated channels that open when acetylcholine binds to them. They are non-specific ion channels. Sodium diffuses into the cell through the channels. A little potassium gets out. It can facilitate both sodium and potassium. The cell membrane briefly becomes less negative or more positive at the end plate region. And the end plate potential is going to be local, but it does lead to the opening of voltage-gated ion channels in the adjacent region of the sarcolemma. And so what that means, if we can go back, the end plate potential occurs here at the motor end plate. It's a change in voltage across the motor end plate. And we are making, when we stimulate these receptors, we are making sodium ions diffuse into the cell, which makes it more positive inside the cell. We can actually make it more positive, but if we don't make it positive enough to open sodium channels, then we don't actually excite the membrane. We don't actually stimulate the sarcolemma. We don't start an action potential. If we don't make it positive enough to reach threshold, which means we don't make it positive enough to open sodium channels down the line, then we're not going to start an action potential. And so this can happen. And so we need maybe sustained stimulation of the motor end plate or multiple stimulations of the motor end plate in order to let enough sodium ions through to make the membrane positive enough to initiate the action potential. Now, when they say it's local, when they say the membrane potential is local, what that means is that the change in voltage just occurs here in the motor end plate. And it's only able to spread on if we can reach threshold and initiate the action potential. Otherwise, the change in voltage just occurs here, and then sodium, potassium, ATPase, sodium, potassium pumps, they'll start pumping the sodium back out and the potassium back in, and we'll get back to our resting membrane potential. All right, so initiation and propagation of action potentials along the sarcolemma and the T-tubules. All right, so an action potential is a rapid rise or depolarization and fall, repolarization, in the charge of a membrane. The EPP or end plate potential reaches threshold by causing, yes, as we said, nearby voltage-gated sodium channels to open. Sodium then diffuses into the cell through voltage-gated channels, and we say the cell depolarizes and becomes less negative. It'll eventually reach positive 30 millivolts. And this results in the opening of adjacent voltage-gated sodium channels and the entry of more sodium. All right, so the chain reaction occurs as depolarization is propagated down the membrane and the T-tubules. That's what we've described before. So just after the sodium channels open, they close and voltage-gated channels will open. So the potassium diffuses out of the cell and it repolarizes by returning back to 90 millivolts. It becomes more negative when we start letting potassium charges, positive charges, out of the cell. The cell will become more negative and we say it has repolarized. All right. While the cell is depolarizing and repolarizing, it is in a refractory period, which means it's unable to respond to another stimulation. All right, and so here we see the parts of an action potential in the sarcolemma. Action potentials will actually look different in different cells. For instance, neurons, the action potential will look a little different. This one's relatively simple actually compared to maybe the neuron, but you see threshold here. That's negative 65 millivolts. All right, and we have a resting membrane potential of negative 90 millivolts. It is 90 millivolts more negative inside the cell than outside at rest. Now, let's say we're at the motor end plate and acetylcholine has bound to acetylcholine receptor. That's going to let sodium ions flood into the cell. If we let positive ions flood into the cell, that will make the cell membrane less negative. If it becomes less negative, then we move up towards zero, right? If it becomes more positive, we move up towards zero, right? So if we let a little bit of sodium in, maybe we don't reach threshold. Maybe we only get to negative 70 millivolts, right? If that happens, then we don't start an action potential, all right? Once we reach threshold, then we open up nearby sodium channels. All right. Once we reach negative 65 millivolts, once we become that much more positive, we open up those nearby voltage-gated sodium channels. And when that happens, tons of sodium floods into the cell, tons of it. And we're going to see the membrane, charge or potential rise to about positive 30. Okay. Now we say it has depolarized. It is going through depolarization. When these charges flip, normally it's positive outside the cell and negative inside. All right. But if we let all those sodium ions flood in, the cell becomes more positive inside, and we say it is depolarized, or it's kind of flipped its polarity. And the muscle cell will reach about positive 30 millivolts. Now, around this time, the sodium channels actually start closing, and at positive 30, the potassium channels will open. Okay, and now the voltage-gated potassium channels are open, and we're going to start letting positive charges out of the cell. We're going to start letting positively charged potassium out of the cell. And we let these positive charges out of the cell, guess what's going to happen when the cell becomes more negative? All right, and so then we go through repolarization. All right, we go through repolarization when we go back to being negative. Right, when the cell becomes more negative again, we are repolarizing. And that happens because we're letting positive charges out of the cell. We're letting potassium out. All right, now that's going to continue to fall. That charge will come down. We'll actually overshoot it a little bit, reaching down here to about negative, 100 or so. We'll overshoot it. And then the potassium channels close. All right. And what's left at this point? Just the sodium potassium pump, which is always running. Okay. It's a little slow, but it's always running. And so over time, it's going to be pumping 3 positive charges out for every 2 it brings in, which over time will make the membrane just slightly more positive, bring us back up to about negative 90 millivolts. And that's our resting membrane potential. And we're gonna stay here thanks to the ATP pump and the leak channels. They'll help maintain this resting membrane potential. All right. So again, if we stimulate the end plate, all right, and we don't reach threshold, nothing happens because we can't open up the sodium channels nearby. If you do reach threshold, you open up those sodium channels nearby, those voltage-gated sodium channels. Tons of positive charges flood in. The cell becomes more positive. All right. Sodium channels close. But now that it's more positive inside the cell, voltage-gated potassium channels open, and they let potassium flood out of the cell, repolarizing it or making it more negative. All right. And then the leak channels and the ATP pump, the sodium potassium ATP pump, will go to work to bring us back to our resting membrane potential and maintain it. All right, that's a complicated little idea. I think the graph does help though. Understand depolarization, repolarization. All right, so the sarcolemma, the T-tubules, more on excitation, contraction, coupling. So the action potential, once we've generated one, right, that's going to travel down the T-tubules. It travels down the sarcolemma. Now it's going to travel down the T-tubules, which are, again, part of the sarcolemma. They travel down into the interior of the cell along the T-tubules. And next to the T-tubules you'll remember are the terminal cisternae and the sarcoplasmic reticulum attached to that. All right, and so this action potential causes the voltage-gated calcium channels to open, all right, in the sarcoplasmic reticulum. And when the sarcoplasmic reticulum releases calcium into the cell, that calcium is going to bind to troponin. All right, I think we should have some better pictures. No, we don't have a better picture, not really. All right, so calcium comes in, it binds to troponin, and when it binds to troponin, it moves troponin out of the way, but I mean, it changes shape and it pushes tropomyosin out of the way. All right. Now, normally, myosin is down here with its binding sites, and it wants to bind the actin in the thin filaments. All right, but it can't because tropomyosin is covering the active sites. It's covering the binding sites. But when calcium binds troponin, it moves tropomyosin out of the way. And now that we have calcium here, and the troponin and tropomyosin are out of the way, now the active sites are exposed, and the myosin heads can bind to the actin filaments. All right? All right. So this cross-bridge cycling is contraction. Okay? Through cross-bridge cycling, we are going to shorten the sarcomeres and cause contraction of the muscle. And again, that all starts with the release of calcium, and calcium then binds to the troponin, which moves the tropomyosin out of the way. Now we get the binding here. All right. All right, so we form the cross bridge, which is where the head of the myosin molecule binds to the active site. All right, on the actin filament. Okay, now we finally got our chance. We're bonding. All right, and bound to the head of myosin is ADP, adenosine diphosphate, and a free phosphate. Okay. Next, ATP, after binding to actin, is going to perform a move called the power stroke. Power stroke or pull on the actin. And when it does that, it's going to release ADP and phosphate. They're gone. All right. Now we're in this kind of completed power stroke position, and we've released ADP and phosphate, and we're stuck there. We're just stuck there until ATP shows up. ATP binds to the active site and actually causes us to release the actin filament. All right. Then we split ATP into ADP and phosphate, and by releasing that phosphate from the ATP molecule, we use that energy to kind of cock the head or reset it. Okay? And so those are the steps. We form the cross bridge. Okay? We're going to release, we're going to do the power stroke next, pull, release the ADP and the phosphate. All right? And then we're stuck in that position until we get new ATP. That ATP causes us to release the actin filament. Now we split or hydrolyze the ATP to go back to the starting position or reset the myosin molecule. Then we bind again, power stroke, release, reset, bind, power stroke, pull, release, reset. And so this can be a little hard to picture, like what's happening when we do this? Why are we binding, pulling, letting go, and then going back to the same old position? What purpose does this serve? Well, picture yourself pulling on a rope, like in tug of war, and if you're making progress, you're going to be pulling back with one hand, and then you switch your other hand forward, and then that hand pulls back, and you switch your other hand forward. You're kind of switching your hands as you pull on the rope, pull it toward you. All right, so if you look at the thick filament, there are lots of myosin heads here on the thick filament, and they're all trying to bind actin at the same time. They're all working together to pull on actin. All right, so they're like a team working on this tug of war rope, right? They're when we release, when one head releases the actin filament, the thin filament, we're not totally letting go of it. There are other myosin heads holding onto it, and so they're all going through this cycle of grabbing, pulling, letting go, and resetting. All right. So anyway, when they do this, they pull the thin filaments toward the M line, and in doing so, they pull the Z disc toward the M line. All right. And so you can see if we pull this Z disc on the right toward the M line, and we pull this Z disc on the left toward the M line, then we've shortened the sarcomere. Okay. We've shortened the length of the sarcomere. This is temporary. Well, let me talk about that in a second. But when we shorten the length of the sarcomere, remember a myofibril is made up of repeating sarcomeres. And so here's a Z disc, here's a Z disc, here's a Z disc, here's a Z disc. We've got several sarcomeres running down in a series, all right, running down the length of the myofibril. And when each one of these shortens, they're all shortening at the same time. And so each one shortens only a little bit, but if they all shorten just a little bit, then we've generated a lot of shortening along the length of the entire muscle, right? Each one only has to shorten a little bit, and if that happens, then but they're all shortening a little bit, then we're going to generate a lot of shortening in the muscle. And that shortening is contraction. Okay, when we contract our muscles, we are shortening the myofibrils. All right. So, yes, cross-bridge cycling will continue as long as calcium is present. Um, if we stop stimulating the muscle fiber, right? If the neuron stops stimulating the muscle fiber, if it stops releasing acetylcholine, and acetylcholine stops binding to the receptors at the motor end plate, we're going to stop producing action potentials. If that happens, the calcium pumps in the terminal cisternae and the sarcoplasmic reticulum, they're going to start sucking up calcium out of the sarcoplasm. They're going to start pulling it out of the sarcoplasm. When that happens, calcium is going to let go of the troponin, and it's going to get sucked up into the sarcoplasm as well. And when that happens, troponin and specifically tropomyosin are going to move back into place, and they're going to block the active sites, and then the myosin heads can't bind to the thin filaments anymore, and so that ends cross-bridge cycling, that ends contraction. When we stop stimulating a muscle fiber, basically it sucks all the calcium back into the sarcoplasmic reticulum to store it for later, and then the regulatory proteins move back into place and just block access to the binding site. All right, so here you can see contraction. You can see the width of the I band, the light band here. This is at rest, and then during contraction, the I band is going to be pretty negligible. The thick and thin filaments are overlapping quite a bit, yeah, in fact, almost entirely. All right. Good. Okay, so we've been talking for a while. I actually want to go ahead and take a break for today. We'll pick this up next time. And if you have any questions, please feel free to email me.